Right, hello Udina, and welcome to the last lesson before the summer holidays, where we're going to be continuing on with our walking talking mock on bioenergetics. So in the last session we went through questions 1 and 2, and today we're going to be going through all of the parts of question 3 and 4. So make sure that 1, you've done the questions, and 2, you've looked at the previous video before doing this video. So we will kickstart where we left off on the last one. So question number 3. Figure 4 shows a single-celled alga which lives in fresh water. So we've got some different structures too here that we should be familiar with. So flagellum, we know those from bacterial cells. They aid in the movement. We've got a vacuole, a nucleus, chloroplast, cell wall, cytoplasm. We're all familiar with those. And then we have a light sensitive spot. Now please be aware that you will never have studied anything like this in your science lessons, but you probably will be asked questions where you have to apply your knowledge to a different situation. And quite often that application comes under assessment objective 2, which again will be worth about 40% of your marks on your tests at GCSE. Question 3.1 though is not assessment objective 2, it's not application, it's just knowing about cellulose and where you find cellulose. So 3.1, which part of the cell labelled above is made of cellulose? That will be the cell wall. So the cell wall in plants and algae will be made of cellulose. So cellulose is the answer. 3.2. Water enters and leaves the algal cell. What is the name of the process by which water moves into cells? Now this is something that we've done in a different topic, to do with transport in cells. So do be aware that there will be a lot of linking between different chapters and even different subjects. So bringing in chemistry and physics into biology questions. So the movement of water, specifically, the movement of water is going to be osmosis. They are, however, very lenient and have given a mark if you have put diffusion, because osmosis is a specific type of diffusion. So osmosis is the preferred answer, but you do get a mark if you put diffusion. Okay, question 3.3. Describe what happens to the algal cell as water moves into the cell. So this is the same with plant cells, which came up in the osmosis section and on the osmosis required practical. There's a couple of answers you could give. The ideal answer is if you've put turgid, goes turgid. That's fantastic, well done. They will, however, also accept swollen. Obviously, the water is going in, so it's going to increase in size, and that is called turgid in plant cells. The cell becomes turgid. The opposite, if water leaves a cell, then it becomes flaccid. Okay, moving on to the next question, 3.4. The alga can photosynthesize. Complete the word equation for photosynthesis. Now, you have to know both the word and the symbol equation for photosynthesis. So having the word equation is the slightly easier one. Doesn't give you any options, but it is worth 2 marks. So hopefully we know that for photosynthesis, it's water plus carbon dioxide, 1 mark, makes with light energy, glucose and oxygen. 1 mark for glucose, 1 mark for carbon dioxide. Now they've put in bold here, complete the word equation. If you've put the symbols CO2 or C6H12O6, you will not gain the marks. If it asks for a word equation, it must all be in words. If it asks for a symbol equation, they must all be in symbols. So do be careful about abbreviating things like carbon dioxide to CO2. You do need the words if it's asking for a word equation. So be careful with that. Okay, 3.5. The flagellum helps the cell to move through water. Scientists think that the flagellum and the light-sensitive spot work together to increase photosynthesis. Suggest how this might happen. Now, I think we've mentioned suggest before. Suggest is an important command word to remember because it will probably alleviate a little bit of stress in your exam. If it is saying suggest, it means that they don't expect you to know that answer, but use your science to come up with a logical explanation, a logical reason, or in this case, a logical way in which it happens. So it's worth 2 marks, so we need to get 2 points in there. Now, if in your answer you have mentioned that the light-sensitive spot will detect light, then that will be worth a mark. If you said that the flagellum moves towards the light, or the flagellum moves the alga towards light, then that would be worth a mark. If you said that more light means there's more photosynthesis, or words to that effect, that also would be worth a mark, but it is a maximum of 2 marks. So the flagellum helps the alga move. The light-sensitive spot is obviously sensitive to light, so it causes it to move towards the light, so it has more photosynthesis. Right, question number 4. This question is about photosynthesis. Plants make glucose during photosynthesis. Some of the glucose is changed into insoluble starch. What happens to this starch? Tick 1 box. Now again, it's worth 1 mark, so please make sure that you are only ticking 1 box if you've been asked to do so. So for this one, always make sure that you read all of the options, because you might think that you know one if you come across it, but there might be one that does actually work better later on. So do make sure that you read through all of the options. So the first option is the starch is converted into oxygen. The second one, the starch is stored for use later. And the third one is the starch is used to make the leaf green. Now, going through those ones, you can already scrub some of them off. So the starch is converted into oxygen. That happens in respiration, where the starch is broken into glucose, which then breaks down to oxygen. So it's not the first one. The last one, we know that it's the chlorophyll in chloroplasts that make leaves green. So it's not the last one. It is the middle one. Starch is a storage molecule, so they store it away and use it for later. So well done if you ticked the middle box. The starch is stored for use later. Okay, 4.2, and we have a practical here. As I've said in previous walking talking mocks, 15% of your science exams will be based on science skills, so carrying out practicals and that kind of thing. Photosynthesis, there was a required practical for that, so those are the ones that they will ask you about. So make sure you always know before your exams in year 11, make sure you know all of the required practicals inside and out. Quite often though, they'll change little bits of it, so do make sure that you're thinking about it as you go. 4.2. A student investigated the effect of light intensity on the rate of photosynthesis in pondweed. Figure 5 shows the way the experiment was set up. So it's very, very similar to the one that we watched on the video in one of the lessons in lockdown, but there are some differences, such as the beaker of water and the thermometer. The student needed to control some variables to make this investigation fair. State 2 variables the student needed to control in this investigation. So before we answer that, first of all, variables. Variables are things in a scientific investigation that can affect your results. We've got the independent variable, that's what we change in investigations, and we only ever change one thing. The dependent variable, that's what we measure in an investigation, so it depends on the independent one. And we have control variables, things that have to stay the same to make the investigation fair, or we can also say make the investigation valid. Now, there's a couple of different answers you can give for this, so I'm going to list some of the answers you might have put. You only had to state them, so the command word is state, so you don't have to explain the effects or describe how you would do it, it's just state two variables. So you could have put carbon dioxide or carbon dioxide concentration. You could have put the temperature. You could have put the light color or the light wavelength. You could have put the pH. You could have put the size or the length of the pondweed. You could have said about having the same species of plant or pondweed, or same type of pondweed. And you could have put the volume of water in the tube. Any one of those would be worth a mark, with a maximum of 2 marks. Okay, 4.3. The bubbles of gas are only produced while photosynthesis is taking place. What 2 measurements would the student make to calculate the rate of photosynthesis? And this is worth 2 marks, so you know that you're going to get a mark for each correct measurement. Now, anything to do with rates, either in biology, in physics, or in chemistry, if it's to do with rate, that is over time. So it's talking about something happening over time. That's what rate is. So one of the measurements that you would need to take is time. So if you've mentioned anything about taking the time, or you've said 5 minutes, doing it for 5 minutes, or 3 minutes, or anything like that, that will be 1 mark. The other mark is the number of bubbles. You can put the amount of bubbles, that would also get you mark, or you can put the amount or the volume of gas produced, or the amount of volume of oxygen produced. But I imagine most people, and I would put the number of bubbles and the time taken. Yes, those would be your 2 answers. I don't know where I was going with that, so okay. So next question, figure 6 shows the effect of light intensity on the rate of photosynthesis in the pondweed. So we've got a graph here, a very familiar graph, where we have the rate increasing as light intensity increases, and then it's plateauing off at C. Excuse me. On the y-axis, we've got the rate of photosynthesis, and on the x-axis, we've got the light intensity in arbitrary units. You might come across the words arbitrary units, it's just sort of general units for it. So the question is, name the factor that limits the rate of photosynthesis between the points labelled A and B on the graph. Now, make sure that you are looking at the right section of the graph, because you might be tempted to do something between C and D, but between A and B, it's being limited by light intensity. Now, how do we know that? Because as the light intensity is increasing, it's going up. So it's not being limited by anything else. Between A and B, it's going to be whatever is on the x-axis. So in this case, the light intensity. The next question, however, says, suggest which factor might be limiting the rate of photosynthesis between the points labelled C and D. Now, there is actually a couple of answers that you can give for this. Obviously, because light intensity is on the x-axis, it's not going to be light intensity, so we have to try and remember the other limiting factors in photosynthesis. So if you have put temperature, or carbon dioxide, or concentration of carbon dioxide, or anything like that, or the amount of chlorophyll, any one of those would be worth a mark. So it's whatever, whichever limiting factor it's not showing you on that graph. That's where it's plateauing. And we've got to remember those four. So light intensity, concentration of carbon dioxide, temperature, and the amount of chlorophyll. Okay, so that is it then from me. We've gone over all of the questions now for the bioenergetics, so very, very well done. The only thing we didn't have chance to go back over and revise and do questions on were the preventing illness, so the communicable diseases section of it. So that might be something that you want to have a look at either during the summer or next year, doesn't matter, but it's something that you haven't had a chance to go back over. Again, I hope you found this very helpful. Getting a lot of feedback saying that people have found them very helpful and very useful in how to answer the questions, so I'm glad that that has helped. That's great. If you could send me your marks for this paper, either on email, the usual address, or via show my homework, then that would be very much appreciated. I hope you have a fantastic summer. I know it has been a little bit weird over the past few months, and I appreciate that you have stuck with it and gone through the online lessons. They're not ideal. It's not as useful. You can't ask questions and things like that you would normally ask in the classroom. But very well done for taking part in them, and hopefully it means that you'll have a better understanding. There is time in your GCSEs to revisit all of these topics, so don't panic if you feel that you haven't understood it as well as you would want to. There is time over the next couple of years to go back and have a look at these sections, and we have written them into our sort of programs of study, so they've been written in when they're going to be done. Great, stay safe over the summer holidays, and hopefully we'll see you in September when we're all back and starting to get back to normal. Take care of yourselves, and goodbye.